Hello everyone and welcome back. It's week 3 and I'm doing part 1 of Bacterial Morphology and Staining Techniques, and it's June 4th at 10 p.m. I'm going to go ahead and do this as quick as possible because it's a long part of the lab, and I did already part of it starting with the staining, with scraping of the gums and teeth. And I already lit up the candle, put over the slide over it, and let it sit for 10 minutes. And so I'm going to briefly go over this really quick. I have the crystal violet, the alcohol, the tap water, everything here, all the items I needed, the bleach, the bucket, everything's here. So now we're all ready to go. I'm going to start now explaining the next step, which is I need to place this inside here with a drop or two of the crystal violet, and I'm just gonna let it stain. So I'm gonna do one, I'm gonna do 2 actually. Okay, let it sit there for a little bit. Meanwhile, I'm gonna go over the technique of the bacterial stain. It's an acid fast stain, and there are 2 types. One's gram positive and the other one is gram negative. So the gram positive is waxy myelic cells, and one of them is called the Zile-Nelson technique, and the other one is the Kenyon technique, and both are the carbonyl fusion, and it's the original stain. Okay, and the gram negative, the gram positive is a thick cell, and it's the peptidoglycan. It's a purple, which again is the crystal violet. And then the thin one, which is the gram negative, it keeps purple stain and it's also pink, is the saffron gram stain, which is number 4. The morphology arrangements, spear-like hocus, and there are 3. I just gave one, the spear-like hocus. The other one is a rod shape, which is the bacillus. And then the spiral one is kind of twisted. It's the spiral one. And then there's a round one, which is the cocci. So the bacteria smears are film surface of the slide, and it's fixed with heat, which I already did that with this slide, with the 1 direct M slide we're using. And I fixed it with heat to do like a positive electric charge. And group, it's how it's stained. The negative part is it keeps normal stain. The colorization or the decolorization is this crystal violet, and it is a thin layer. And we're going to use the microscope as a way to visualize cells and parts. Okay, so now we're going to move on. After I did that, I gently rinse the slide with tap water, holding it at an angle. So I'm going to run a little bit of the tap water. I'm going to hold it at an angle, just to pour a little bit. Okay. And I guess I should put a little bit more. Okay, so what you want to do is you want to hold it at an angle to run just enough to hold the slide, because I let it sit for 60 seconds. And then I'm going to take one of the paper towels and kind of wipe it off a little bit, so we can use this under the slide. And I'm going to dry the slide with paper towels after staining. And I'm going to place the dried slide up under the microscope using a low, medium, and high magnification. And so we can see what the cells will look like. Unfortunately, I'm not going to do it on this segment of the video because it's going to have to have more time, and we only have 5 minutes. I'm going to take photograph with this and then place this for the lab actual report. Okay, and the magnification I will be recording on my data. And so the second label I have done already, is the -2M slide, and I'm going to place that on here as well. So I'm going to get that one. And what I'm going to do is I'm going to make a circle as well on the back with a permanent marker. And on the back of the slide with a permanent marker. And I'm going to do this one more time. I'm going to actually use the Congo one, which is this one. And I need to kind of wipe this out a little bit so it doesn't and I already cut off the top of the Congo, so that way I can already start this next staining. And I'm going to stain the negative slide in the middle. Okay. And you only need to put a drop for this one. Okay. Hold on 1 second. Do it one more time. Hold on 1 second here. Okay. And you're going to do the next step, which is you're going to put everything, the stain, you're going to take it from the mouth. Negative indicates procedure and the stain M indicates sample to be taken from the mouth. Okay, I already did that. So what I want to do is I want to stain the area. So hold on, let me do this again because for some reason it didn't take. Oh, here we go. Okay. So then I'm going to go ahead and do that again and gather the second sterile swab. I've already did the teeth and the gums, and then place it on the Congo red stain and sample the transfers to the swab stain. And then I'm going to let it locate a clean blank slide and place the slide at a 30-degree angle to contact and spread the dye across the slide. Okay, so I'm gonna do that segment after the video. And then the next one, as I need to let that completely dry, so I'm gonna do that in the next video. And then I need to do the one for the -3. So now I'm going to do the same thing, and I'm actually going to put on the they want us to, okay, so once I do that with the slide, when I move it around onto the 2 negative M, then it will I'll be able to place it underneath the microscope. And for this segment, that will be it for this segment, and until next time, thank you.